Hi, everyone. My name is Bill Daniels, and I'm the program leader for Native Seed Communities, which is a project of the Indiana Native Plant Society. We're very happy that you've joined us tonight for Melissa Spearing's presentation, Waking the Spring Beauties, Boost Your Success and Local Biodiversity. Before I introduce Melissa, I'd like to share with you some information about Indiana Native Seed Communities. We promote networks of native plant enthusiasts working together to procure, process, and propagate regionally native plant seeds to increase the presence of these beautiful and wonderfully appropriate native plants in all of our landscapes. Our presentations are devoted to discussing the propagation of native plants from seeds and the spotlighting of community organizations and individuals that mobilize native plant enthusiasts to use these seed-grown plants in all of our landscapes. We have a lot of resources on our Indiana Native Plant Society's website, as well as we've got the various social media opportunities, including our Facebook group, Instagram, and we invite you to share your pictures and other experiences, especially what your communities are doing in our Facebook group. For the best experience for everyone, if you would please keep yourself muted unless you're invited to unmute. Also, if you have any questions, we will be taking those questions in from the chat. So put your questions in the chat, and then at the end of the presentation, Melissa will be fielding those questions with the help of Deb Housen, who's also helping with various technical aspects, etc. Okay, well, let's go ahead and move into introducing our speaker tonight. I'd like to now turn it over to Ray Major, who is a Native Seed Communities Committee member. He's also, and many of you may know him, the editor of a popular Facebook page, Trees from Seed. So thanks, Ray. I'll go ahead and turn it over to you right now.
Thank you, Bill. This is a great pleasure. Melissa Spearing is a professional seed biologist helping to steward over 15,000 conservation seed lots at National Tree Seed Center in Fredericton, New Brunswick, while working towards a Master of Forest Science degree at the University of New Brunswick. Her background spans nursery production and scaling up grow me insteads at her parents' farm in southern Ontario, advancing species at risk recovery programs across Canada, delivering seed tree collection training for the Forest Gene Conservation Association of Ontario, training at the Millennium Seed Bank in England, and Melissa holds a Niagara Park School of Horticulture diploma. Her passion is helping growers to preserve and all the native species and to help them thrive in advance of a rapidly changing climate. Melissa, it's a pleasure to introduce you.
Thank you very much, Ray, and thank you, Bill, and thank you all for coming tonight. And yeah, I realized that was a mouthful of a biography, so I'll make it more plain language next time. Yeah, so am I good to share my screen now? Okay, so okay, you guys confirm if you can see that?
Yes, we can.
All right. Okay, still good?
Yes.
All right, wonderful. Okay, so yeah, I can talk for a long time about a lot of plants. And I said to Bill when we were chatting about this that it's really nice to sort of have a focus in terms of all the different things I've grown. I mean, my personal joys in life are trying to grow as many different things as I possibly can. And I'm so lucky because I had my parents my parents are just such green thumbs, and so I learned quite a lot from them. But I've also been so lucky in all the educational things I've gotten to do. So a lot of this is sort of my old life, I'm going to call it. As Ray said, I'm doing a lot of work now with forest conservation. I am going to talk about some special trees that I consider in the same boat as a lot of the woodland ephemerals, but I love having so many photos because I've gotten to know a lot of these woodland plants. And growing up in Southern Ontario, it's probably quite a lot like Indiana in a lot of the species that are shared. And also a lot of the same conservation issues in terms of fragmented habitats and pressure on woodlots and whatnot. But I will try and cover everything. And yeah, I'm really open to having lots of conversation at the end. So I'll try and get through this and see what sparks your interests. And we can talk about all sorts of species that I just may not have found the photos for. So yeah, I would like to acknowledge that I am speaking today and tonight from the unceded territory of the Wolastoqey people in Fredericton. And it's the home of the Peace and Friendship Treaties. And so, I get to do a lot of work now and travel a lot of different places in North America. And it really is nice to see the reconciliation movement moving towards recognizing all of these territories that we've taken over or cross or utilize or are just lucky to live in and work with. So I get to work with a lot of Indigenous groups and seed conservation as well. So it's been a really wonderful part of the current work that I do. But I really do understand now that if we're working on healing the land and doing native plant gardening, it's just one thing that you can incorporate into your life that'll sort of heal the relationship with your local sense of place, the local peoples, the local names. And one of the things that I keep hearing about all the time with Indigenous people is that the more people get into native plant gardening or native, just learning to understand the ecology around them, is that you also help preserve their languages, because you'll inevitably get possibly into reading about or hear different names for things instead of the Western names or the scientific names. I mean, I'm very much on the botanical Latin side in terms of my training, because it's been very traditional botany, but I'm starting to want to learn. I try to learn 1 or 2 Indigenous plant names a week now, if I can. And every one of these communities and these nations might have a different name for a different plant. And I've heard really cool stories from, even Western Canada, where buffalo berry that grows on the top of the hill has a different name than the ones that grow in the water, so, or along the riparian zones. So there's all sorts of really interesting things like that. So yeah, as Ray said in my bio, I grew up in Southern Ontario. This is my parents' nursery that's in Bethany. And so my parents got started at growing perennials in the late 70s. And at that time, there were almost no growers of perennials in Southern Ontario. And, it obviously gardening's evolved. I mean, it's what I like to call the carpeting of the horticultural world. Is, back in the days, it was the daylilies and the hostas and the ivy and the vinca, and those things just slowly started evolving. So, and my dad was also one of the first growers in Ontario that really started working on a lot of cottage country plants. So things like bearberry and cornus and gaultheria, and sort of trying to come up with enough plant material at a large scale. And I mean, we are growing, millions of plants some years to produce. So originally it was a lot of clonal production, and then I started really getting interested in seed production. So all the native plants that we've been slowly trying to convert them. So I'll talk a lot more about that. So this farm was an absolute barren field when we started, and a lot of it was about building the soil up. It had been fairly denuded, digging the pond. So this is me when I was a little kid. I think my sister was like just born in this year or two. So the front field had, pretty much nothing in it to begin with. And so I grew up as things got built up. So it was a really fun place to grow up as a kid. I'm very appreciative now of how wonderful that was, and how much it changed the course of my life. And from there, this is Niagara Parks School of Horticulture. I got to take this photo in the bucket truck because I had a really good camera when I left my job in marketing in Toronto. And again, my parents both went to this school, and then I went there, so I've kind of been fully infused. And this school started to open up my world because I did a seed exchange program. And so I was collecting seed for other gardens around the world that wanted to grow plants from the Niagara region. And we traded seed, and that's how the seed exchange sort of bug got in me. And I started learning about all these different things. The tree conservation stuff really started happening when I got asked to do some seed collections for ash conservation. So this is a blue ash seed orchard actually at the University of Guelph Arboretum. And they've had a long-standing program for seed-based restoration and seed propagation, but also connecting a lot of disjunct populations that are hundreds of kilometers apart that wouldn't normally breed in the wild. So 2013, this is the first year that I helped send some seed to the seed bank I work at now. So, you know, sometimes you wait years and years and years to get an opportunity to work with the plant. And this was a really amazing opportunity. So I started doing a lot of ash conservation work around Toronto when the emerald ash borer hit. And that was a big part of it. And then in 2014, I was really lucky to get accepted with representatives from 14 other countries to go to the Millennium Seed Bank. And so I lived in the Millennium Seed Bank for 3.5 weeks. And we had classes all the time, and we did all these wonderful lessons with everyone. And the year after that, I helped publish what had been the province of Ontario used to have a seed plant, and they used to have all these programs for paying seed collectors for their bank going back to the 1920s. So we redid the manual to help with seed collection training. So I really got to infuse everything from all the pictures on this slide here into like a great book. And that I get so excited when I talk to people in the Yukon now, or people from, you know, the Lake States in Wisconsin, and they on the Zoom call and they hold up the book and say, this one's great, it's got lots of photos. And that's what I think a lot of people need these days. They need pictures, and that's why social media is so wonderful. So yeah, so how it's going now. I am a bit obsessive with seeds. I feel like it's pretty much everything I do. I love how much you can adapt and multiply. When you get good at this, when you get to know a plant, you really start to understand how, you know, what makes it happy. And this bed behind me, these ginger plants, like this was the first year that I had 60 or 70,000 seedlings of Canadian ginger available. So, you know, my dad started challenging me to say, well, if you really want to, you know, replace Vinca, or if you really want to do that, you're gonna have to get real good at it. So he also has a saying that I really love that's you know, it takes 10 years to know a plant, and it takes 10 more years to grow a plant really well. And especially when you're, you know, in it for money, I guess. I mean, to pay the bills. But, you know, the whole farm experience, the whole thing becomes obsessive when you're starting to think, well, I can grow my own food, I can grow my own herbs, I can grow fruit trees, I can go foraging in the woods, and, you know, I go pick mushrooms because at the same time I'm going to get trillium seed or something. So it's just kind of wrapped in everything. I think seed is just a metaphor for most of life. So now I get to do really amazing work, and I get opportunities to help forest restoration companies. I help the 2 Billion Tree Program, and I do a lot of, or I help manage our seed bank here, which is growing by leaps and bounds. And yeah, just a postscript, I've never been to Indiana, but I want to come now. So yeah, I'm not going to talk too much about work because I'm doing this on my own time, but just the National Tree Seed Center has, we do seed exchange with researchers all over the country. We also hold seed, again, for ash or for butternut. So the picture up in the top right here is butternut embryos that I was helping cryopreserve the first time I ever came out to the seed bank in 2016. But most of our seed bank is of species that we call or know to be orthodox, so they can be dried and frozen for, you know, decades. So we've got some seedlots that are still germinating from the 1950s, from all over Canada, mostly pines and spruces, because that's what the forest companies were interested in at the time. But we are starting to move into a few more challenging species. And so the best part of my job is researching things that, say, Indigenous groups care about, because to them, the forest isn't just the trees, it's also all the plants that live below it. So I get to use some of my knowledge about woodland ephemerals in that way. So yeah, so I'm gonna try and get through this in about 30 minutes, but so yeah, Bill and I had a good chat, and I apologize because I did have a bit of a technology issue and I did lose some slides and material that I'd worked on, but I'd be happy to keep working on things and send them to you guys later on. So yeah, I want to talk a little bit about what makes spring ephemerals so special to me, to you, to anyone. I know how appealing they are when they're in flower, but they are incredibly difficult to grow. And I think the biggest thing I can say now is going to be about patience. I'm going to show you guys a few little seed challenges and tricks. I asked Bill for his choice of plants, and he picked wild geranium. So that one's I've got a few videos to show you from my seed gardens. And then I've got two special trees to me that are kind of the same as woodland ephemerals in terms of how hard their seed is. So yes, what makes woodland ephemerals so special? And I already alluded to it a little bit, but they are just slow. They challenge everybody in terms of the nursery sector, in terms of getting to know them and understanding what they're doing above and below ground. And one of the things that I feel fortunate to be able to do just because of nursery production is that, we have to pot and repot things. And one of the biggest challenges I had getting going with any of these plants is just knowing that when they're dormant is the absolute best time to move them. So, in the summer period when they're dormant or they've lost the roots and there's nothing above ground, that is the best time to relocate something. For most people, most landscapers, they want to move things when it's in full flower. The retail garden centers want it on the racks. But, truthfully, if you want to get good with these plants, you need to know that there are just very opportune times to get them to go before they put on a lot of roots. And before their seed pods, they're taking all of their energy in the springtime to flower and produce seed as quickly as possible before the trees leaf out. So it's really important to leave them alone during that period if you can. The other thing I was going to say too is just how long it takes some of them to size up when they're not in the best soil. So a lot of them don't grow very quickly because it tends to be garden soils aren't always the most nutritious. So I'll show you guys some really interesting things that I've learned with rotten sawdust, of all things. Has been my best kept secret, perhaps. And yeah, I just want to start off too with a little sort of picture about, people think, why are they so special? Because they're so difficult to grow on mass scale. There's not a lot of growers that produce them. And so the ones that are in trade often come from perhaps tissue cultured in Europe, or they come from, unfortunately still there's a lot of wholesalers that dig things from the woods of Tennessee and sell them online for very cheap, and growers can just buy them and pot them and do a quick sale. But, the fact of the matter is sometimes the trilliums you might see on a rack at Canadian Tire or some of our other garden centers, they could be 10 or 20 years old because they've come from the woods. So there's been a lot of studies done lately, and I've been doing more academic work. Not muted. And so, between a lot of the Google Maps projects, so this is Rick Gray is just he's an author now, but he compiled this all by himself for fun, I think, as a retirement project. We also have the Network of Nature in Canada that has a great native plant map and where to buy things. And it's, if we could get down to filtering this out as to who has woodland ephemerals, it would be a fairly short list. And again, my inspiration came a lot from starting to realize around 2014 that the province of Ontario was probably or possibly going to start regulating things like ivy and vinca. And for my parents, that was just a risk that we needed to diversify. So I went to the woods and I started picking things that I was allowed to pick. And that's a very important part of it. But, around where I live, there's a lot of intact woodlots, and a lot of our neighbors and whatnot didn't mind me poking around and trying some things. So I started at Niagara Parks because actually the woodland garden, my teacher, Lauren Fass, joking thing is that, you guys are so lucky in the US because I think there is such a robust native plant group, 841 vendors from the last study I saw, up to 74% of the Midwest species are available. And, you can give Prairie Moon a lot of credit here because they've just been growing in leaps and bounds. But if you extrapolate that to Canada, I would highly doubt that we have 1,540 species available. And of our woodland ephemerals, there might be a handful that people can buy, kind of most of the time across most places. So I think it's really important and nice to see that more people are getting on board with this. And I just did a podcast with one of my colleagues, Dr. Stefan Weber, with the Native Plant podcast that Pinelands Nursery runs. And, I just sort of talked about, or we talked in general about the state of native plants in Canada is growing, and it's fantastic, but we don't want it to take away from, hopefully, don't take away from the people that have been working really, really hard in the background for 10 years to bring something to the market and in a sustainable way. So I'm really going to emphasize just that we want to try and do this in the most sustainable way we can. It's not fast. So Bill did give me a few resources, and I think it's really wonderful. I just want to talk too about collecting your own seed. I always get permission no matter what I do, because it's just not a good policy to give seed collecting a bad rap. I hear one of the things that we teach with our seed collecting courses that I think is it's very in line with a lot of Indigenous views, is that if you don't pick the first, you won't pick the last. And it's a bit of an honorable harvest, sort of. Robin Wall Kimmerer might say something similarly in her books, but the whole point is that if you care about any of these species and understand how rare they are, a lot of the plants that we really desire are rare because they've been over harvested, or they're just simply habitat specialists. And most woodland ephemerals have fairly particular habitat needs, and the more they're disturbed, even in logged forests, there's not often a fantastic representation of the ground layer just because of that disturbance regime. So most of our plants, and the ones I'm going to talk about, they don't love disturbance. So if you have a large population of them, you're very lucky. And it's really nice to see the 5% rule, and making sure it's hard sometimes to look over 5 acres of something and say, well, what's 5%? If you saw some of the amounts of seed that I've picked at times, you'd say, well, that's a lot. But my brother has 7 acres solid of, wild leek. So if I pick a pound of seed, it's not going to go amiss. But I've been very careful and very deliberate in trying to make sure that I don't constantly return to the same places. Also, is that I have a strong network of people that I can reach out to and say, hey, do you mind if I come to your place this year? Because the last 2 years I had to go to the other spot. I also have a rule about, sort of every 1 hour's drive north, you kind of, in phenology, go back a week. So if I miss something, and if I would miss something in Niagara, I would drive an hour north and have another opportunity or another population I could go to where I was allowed to pick, and I could spread my timing out as well. Yeah. Oh, and Adlumia is on here. So my plant that I still have never collected seed of it. So Adlumia fungosa is a climbing bleeding heart plant. It's so beautiful. It's one of the ones that's kind of like it's my unicorn plant. I've only ever seen it in the wild twice, and I almost wanted to pick some, but I was like, no, I can't. It's just too small. So yeah, again, why so special? So woodland ephemerals are special because I think that they represent some of the most evolved and complex seed types. And I'm going to go a little bit basic biology here, but if you've never heard of Carol Baskin and Baskin, they're husband and wife couple from the University of Kentucky. They've written the Bibles on books. This book in particular, the update that came in 2014, has over, I think, 15,000 species categorized in it. But the thing that's amazing with what they've done is that they've sort of been able to take the entire world's seed types, and they've condensed them down really into 5 categories. And it really starts to help you think about, what's an easy species, what's commonly available in trade, how do I organize my winter sowing, or how do I, which ones do I need to pay more attention to? So, in general, all of our non-dormant species in the world, so most of our vegetables, most of our food crops, they're non-dormant because they evolved in areas that don't have much of a season. So they are pretty much ready to germinate the second they have a fully developed embryo. They typically germinate quickly as soon as there's water. And those are, that represents about, I think, a quarter of the world's species are from areas like that. So without a winter. The second that you go towards the poles, you start to have physiological dormancy. So mom plant tells the seeds, you're going to experience some cold, dry, not great conditions to germinate in. So I'm going to give you all the food and the package that you need, but I want you to stay asleep for the winter. So a lot of those species that we do cold and moist stratification on are Bill's wonderful germination stations. Winter sowing is all based on breaking physiological dormancy, because most of the time you just need enough cold to degrade some abscisic acid. And once that's gone and then the temperatures warm up, they'll germinate. So I'm going to skip to the very bottom though, because you guys would also probably be very familiar with species that have physical dormancy, where all you need to do is scarify the seed coat. So your Kentucky coffee trees, your legumes, your lupins, it's just basically a plastic bubble. You just need to sort of scratch that seed coat, and then the water can get in, and then they're essentially non-dormant. But the last two, you'll see a lot of this through this thing. So I've given them the little code. So morphological dormancy and morphophysiological dormancy are where you have underdeveloped embryos that need cycles of warm and cold to grow first before they can even wake up. So the ratio of the embryo to the seed is lots of times a clue about why species are challenging. And most of our woodland ephemerals are in these two M categories. So, so Baskin also has some wonderful things. So I don't know if you guys do it, but I do a lot of cut testing at work. When we're doing seed testing, the first thing I start telling people is there even an embryo in it? You have to know how to look, where to look. I've got a really great resource I'll show and share with you guys. And obviously trying to get a sense of picking seed at maturity. What color is it? Are the pods splitting, like that picture in the hand with the bloodroot I did? If you pick bloodroot when the seeds are white, they don't germinate as well, and they're very at risk of molding early or being damaged while you're handling them. So you want to pick seeds that have fully matured, and especially woodland ephemerals, because the embryo is already undersized. It's already a bit of a premature baby. So if you get it when it's underripe, it doesn't help. A lot of this has to do a little bit more with lab experiments, but even with woodland ephemeral seeds, use them right away. Don't put them in a drawer. You want to emulate as much as you can what would happen in nature. The trillium seed pod falls off, and literally within 10 minutes or an hour, the ants are walking away with them. So you really want to deal with these as quickly as you can. And we do a lot of testing, so I'll show you some testing. And again, if you're not sure of what a species is, if you just nick the outer seed coat with like a little blade and see if it swells when you put it in water, that's usually a good indication of a species that might just need to have a little crack in the seed coat. I always tell people to think about the weather patterns where you are. You want to, and wherever you're buying seed from, is think about how to mimic the seasons where it's coming from. That's always a fantastic idea. And again, it's typically why a lot of people get really good at winter sowing, because you're essentially following what the plants would do anyways. This is a little bit more about other species, but we want to understand if the seed structures on the outside are causing dormancy. So trying to get down to what we would call pure live seed. You want to get as much of the outer material off as you can, because sometimes that's an impediment. And if you're buying seed or if you're trading seed, know, especially with ephemerals, and I've done this several times because my dad would go and buy seed from he bought I remember him buying years ago wild ginger seed from someone that I think we imported it, a little bit of it from the States, and it was completely dried out. And I was like, Dad, we can't, it's probably dead. Nothing ever came of it because it just can't be dried out. So that's when we just started turning to our own sources. So yeah, again, why so special? So these, I really love this. This is 1946, these little black and white drawings. There's a huge paper that was done, and it was someone just drawing very simple diagrams of seeds and then showing where the embryo is and how, so that little black dot is typically the size of the embryo in the magnolias. It can, like, that little tree can fit through a pinhead. And the first time I had ever done work with magnolia, I didn't really realize how small they are. You know, you see a drawing and you think, oh, I can find it, but it's white and the endosperm is white. So it took a little bit of digging to discern where the baby was. But all these other species, you know, you've got Mediola virginiana, which is cucumber root. You've got red trillium there. That's that little tiny black dot is the embryo. Panax is, sorry, I'm doing the translations in my brain, is, oh my goodness, wild ginseng. There's the wild ginger embryo, that tiny little speck at the bottom, and Adlumia fungosa, the wild bleeding heart. So depending on which way you cut the seed, sometimes you'll never see it. But if you cut it in a certain way with a really good microscope, you'll find it. And I'll say too that Bill Kalina's book, The Growing and Propagating Wildflowers, has fantastic information in the appendix in terms of especially warning people if things can't be dried out, that you need to keep them moist all the time, right through sowing. And his books taught me so many different things. I'm hoping I'll actually get to meet Bill very soon because I live closer to him now. Oh, I love this Facebook post. The one day someone said this is the ant snack pack. And yeah, this is the other part of it. And instead of just the drying part, because that'll reduce germination on a lot of our ephemerals quickly, you know, I've really found it challenging with the fact that all this plants I want to grow, and when I sow, you know, when I sow thousands of woodland ephemeral seeds, that I really had to pay attention to what the ants were doing. And I had to move them around to kind of not confuse them, but, you know, I would sow the bloodroot and then an hour later, there would just be a trail of ants carrying them away. And I was like, I just went for my entire weekend seed picking and now they're walking away with them. So it does make it more challenging when you're working with woodland ephemerals to try to achieve what you want to achieve and have to accommodate the ants will take some. But one really fantastic trick that I learned from Krista Wallach, who I believe is on your guys' website, the Wild Plants from Seed. Crystal is an amazing woman and dedicated her whole life to seed, to selling wild and native seed. She's one of the OGs, like Ray. But Crystal told me a really interesting thing is that you can let the elysomes dry out just enough, but not kill the seed. And also with some of the peroxide tricks that I've been doing is that you can kind of reduce the, I think perhaps it's the smell or the sugar content of that elysome. And you can try to eliminate some of the attractants for the ants so that your seed stays where you sow it. If it's really highly prized seed, but I always let the ants take a few. So yeah, I won't harp on this too much. I just wanted to say, you know, it's interesting because I think all native seed, whether or not it's orthodox or dried out, I mean, I do think a lot of people have better luck when they sow things fresh. A lot of things go more dormant when they dry out. So even though most of our ephemerals can't be dried out, I'm not saying all of them, but a lot of our orthodox seeds, you know, it is good to sow these. And if you want to get better at propagating, is collect your own and sow your, collect it, clean it, sow it relatively quickly for the best success. So so this was my process for a lot of our ephemerals at our nursery. So I would go collect the seeds. So right now these pictures are of wild ginger, and we had these flats that my dad bought. So my dad likes a lot of older school techniques, and we realized that using the plastic flats weren't that great because the water would, you know, when the media would dry out, it kind of pulls away from the edges. So my dad built all these Eastern white cedar boxes, and they have actually mesh on the bottom, and then we would put mesh on the top after we sowed things to keep the mice from tunneling in them. And I would also put cardboard on top after sowing so that all of the flats would stay really nice and moist through the summer. So my timing would be like this. So this is bloodroot and wild ginger are ripe around the same time. So at the end of June in Peterborough area, and we had all these nursery beds or seed gardens that I planted underneath the locusts in front of my parents' house. And I really like underplanting native seed gardens of ephemerals underneath honey locust, because in particular, one, the leaves are really good mulch, and two, they leaf out very late. So the plants got an extra boost of time to finish all of their seed work and flowering underneath these locusts. So that was a really nice sort of accidental discovery that I figured out. But we would be collecting the seeds, we would pick the pods, clean them. I would keep them in Ziploc bags, freezer bags, cool in my basement, not in the fridge. This was something else that I learned for a long time, is one of the tricks was to not put woodland ephemeral seed that thinks it's going to be getting warmer into July in the fridge. I think that also is a little trick, is that I would just keep them, on the cold basement floor in my parents' house for about a week and a half until we were ready and got all of our flats ready. The soil that's in this, so my dad, we've never grown plants on the sort of, a lot of nurseries have either concrete floors or they'll have landscape fabric put down so they can put their pots on top of it. But almost all the things that my dad grows, the ferns, a lot of our woodland plants, and most of the regular nursery production that we do, we have rotten sawdust. So we would get this from a local mill, and it's sort of coarse, well, a ½ inch to ¼ inch chips, and it's mixed deciduous and conifer where we are. And I started to learn for a long time that it would always go underneath in all of our hoop houses, and then it would slowly rot down over a few years. And once it got rotted, it was essentially just like pure humus. And so rotten sawdust, if you could, have a place where you could keep it sort of weed free and just let it mature, it was crumbly, it held a lot of water, and it just felt like the richest chocolate forest soil that you could feel. And it was so much better than regular potting soil. And we had mushrooms that would just propagate themselves. My dad always said we had too many mushrooms growing in the compost because they were coming naturally from the sources that we got them from. But we would sort of fill these trays about 2 to 3 centimeters with soil. I would sow the seeds and then put another 1 to 3 centimeters of that same rotten sawdust on top. Then I would put I'd water it in, put cardboard on top, then put the mesh mouse guard on top of it. And the next spring we would lift them up and see what was germinating. And if nothing really wanted to germinate, I would just keep the cardboard on for another year, because some species just take 2 years to wake up. So it was a really great system. And it's, we would have entire hoop houses like this with hundreds of thousands of seedlings growing each year. So we would always do the transplanting in August or September, and that would give them enough time to put on a few more roots before winter, and then they'd be ready for the spring. So I'll show you guys. No, not perfect. I'll show you guys just sort of how it went year by year. So I started, I had to challenge myself. I wanted to grow trilliums. I really don't love the fact that a lot of trilliums are dug out of the wild for sale. So, the first time that I ever picked just white trillium. So again, my brother's woodlot has so many trilliums in it that I could pick to my heart's content. But the first year it failed. I mishandled the seed. It went moldy on me. I didn't let those elysomes dry out enough. And when they have so much sugar in them too, they just get moldy really easily. So I did a lot of Research. Again, Bill Kalina's got a really great native propagation guide. And so I was looking for different techniques. I talked to a few growers about it, just sort of how people set up and did what they were doing. I did another seed collection. So the picture here is of wild ginger, but I always use 3% hydrogen peroxide now, because I find that it is a great, it enhances germination. At the same time, reducing mold. And it seems to have an effect on the elizome. I don't have perfect scientific evidence of that, but it's commonly available in most oh my goodness. Sorry, I'm stumbling my words here. It's commonly available and easy to get and very safe to use. And then, yeah, 2 years later, I transplanted these tiny little guys with single little leaves into some larger outdoor beds with more of that rotten sawdust. They grew slightly bigger, slightly bigger, slightly bigger. And then I just kept spacing them out a little bit. And then by 2021, 2022, we were able to start selling them. So the picture, the Instagram photo that's down there was from 2022, and that was for sale in Toronto. So it was really cool to see these suddenly flowering on their own after about 5 years, 6 years. So I realize I'm getting late on time, but I'm gonna just get through the end of the geranium talk, and then I think perhaps I will maybe skip over the trees unless Bill tells me that I got more time. But one of the things that I also really love about the woodland ephemerals is learning sort of about what pollinators are really doing. Like when you start to do seed production at a scale, or you're really interested in a particular plant, I spend so much time just watching them when they're in flower, not to appreciate the flowers, but also just because you start to learn really interesting things about what's actually happening. So
I'll have to edit these together. So this is,
I have a video here.
This is a wild geranium, Geraniaceae, and the pollinators love it. The honeybees go after it first, and a lot of the early flowers, I find that they don't set seed very well. And some of this may be due to a cold spring, but this, I think, sometimes the honeybees go, there's nectaries to attract honeybees in here, so the honeybees actually go in behind and steal from there, and they don't touch the pollen, so then these seeds wouldn't germinate. So these are 5, if you can see it closely, 5 little pistils. So 5 little pistillate flowers that connect to 5 ovules. So 5 ovules turns into these. So 1, 2, 3, 4, 5. So after the petals fall off, these enlarge. They happen really quickly. And then as they start to come closer to maturity, the beak goes from, they start to go from brown, they start to brown up, and then they go down to the seed pod. And then they begin to lift. And the lift is really important to watch for because it means that the seeds mature, but it's before they actually disperse. And these are really cool because they shoot up. I'm trying to find one that's totally open. So I've been picking these, and the trick with this species, and here's a couple. The trick with this species is that you need to collect them before they slingshot across the yard. And it's why there's so many little geranium plants scattered from last year, because they shot up from here. Oh, there's a pretty one. They're kind of beautiful. So this is why they call them a crane's bill, because they look a bit like a bird's beak, but when they fling open, they're projectile. And so actually there's a bunch of empty seed pods. So that little space in that little boat is where the seed would be if you got it in time. So lots of times when people learn about seed collecting, they're collecting fruit structures. They're not necessarily collecting seed at first, because if you don't spend a little bit of time dissecting it, you're not sure where you're going to get it. And I actually think now I've got a predator or a bird that's figured out what I'm doing, because it's actually going in and picking these before they're actually ripe. So I'm trying to get the last few that are turning brown today, and I put them in paper bags, and then I let them disperse. And the thing is, in the wild, I'd have to do a lot of collecting and find a really good patch to find just this many in flower. And this is kind of a perfect high canopy, although these trees, these black or honey locusts, took a bit of a beating last couple of years, so they got pretty thin crowns, but the high canopy and light, and we can water these, they get, you know, a little bit of fertilizer or compost every year, really adds up to being pretty good for commercial production, because it's not a plant you can, you know, get a lot of easily by any other means. So yeah, and these are actually 3 different sources. So there's some local, that patch came from Norfolk County, Haldeman Way, and then I've got a few more plants that I dotted in from Toronto. So there's 3 different unrelated families in here. So that is that's Geranium maculatum, and I'll take another clip when I go to do the seed cleaning and sowing and whatnot.
Okay. How am I doing for time, Bill?
We are about 3 more minutes or so. We haven't had a lot of questions, I don't think. But we've got a little more time. If it will be recorded, if people can't stay past 8 o'clock, then the recording will be there. Hopefully, so far it looks like it's recording well. And then it would be nice, since pawpaw is a really popular tree here. Maybe you could do the pawpaw.
Okay. Yeah.
What do you think of that?
Yep. No, that's good.
Okay. Great. Thanks.
Okay. And thank you, everyone. I like I said, I enjoy talking about this so much, and it's fun to get asked. So yeah, as you guys saw in that little video, I mean, I think I covered a lot of that, but I find it amazing at times to just spend enough time learning about these plants and reading about them. There's actually a really amazing paper on the ecological life history of Geranium maculatum. It's from a nun in that same American mid-naturalist, but she was from South Bend in Indiana, and she spent 5 to 10 years just observing and recording and figuring out how Geranium maculatum propagates itself and sort of what happens with the seed pods. And so there's so much amazing literature out there. I think it's often that we feel that there's just, oh, I don't know, I'm going to go ask the hive mind on Facebook. But again, I think if you've had any scholarly training, if you can get into a place that lets you behind paywalls, you start to really find a lot of interesting stuff. And a lot of these old papers have really fantastic little pieces of information in them. So I've learned quite a number of things about Geranium maculatum. I would say in particular that also because it's projectile in the wild, when you're doing seed cleaning, I have to be very cautious when I'm cleaning it, that the pods have all actually opened up, because lots of times, I'll show you this little video quickly. This one's much shorter. But it, I lose seeds in my mom's kitchen all the time, because when we're cleaning it, they're projectiling even off the screens. But one of the things that became problematic for us, and I don't know if anyone else has ever seen this, and I've been asking other people if they've ever noticed it, but in the middle picture here is that a lot of my plants from the video, that huge seed garden that I had, I mean, a lot of them just slowly died out, I think from a virus, or it was a phytoplasma. The flowers would get all messed up, and then they stopped producing seeds completely. So I've never noticed this before. It'd be interesting if anyone else has seen it, but these people tend to think native plants are without problems, but they're affected by aphids and all sorts of different things. And there's not a lot of people doing a lot of research on native plant diseases of herbaceous perennials per se, but in trees, yes, but not necessarily in a lot of the plants that we want. So I'll just show you guys this quickly. So seed cleaning. So yes, with wild geranium, so as soon as the bloodroot and the ginger is done, and the wood poppy is all around the same time for sowing, like once we finish all those big flats of sowing, it's usually when the geranium starts to pick up. And geranium, you have to go out and pick it every single day for about 3 to 4 weeks. And I would cycle through having paper bags with the date that I picked them on, or that our staff picked them on. And so I'd let them dehiss in a, very loosely on their side paper bag. Again, not really cold, and not make sure that they're closed, because they will shoot right out of it. And then I would take bags of the sort of seed pods and I'd crush them. So I have these, I bought them from a medicinal herb place, I think Mountain Herbs out in, I bought these screens, and then I made my own little seed cleaning sieves. And there's not a huge variety of sieve sizes, but they work really well. So I'm always using gloves with geranium. I've hurt myself so many times with the crane's bill, stabbing myself in the hands. But so I just sort of crush it up, and then I would use a fan and just winnow all of the loose dry material off. And So geranium is not one that, so it has a really hard impermeable seed coat. And so it can really take a good rubbing to get the outer shells off, the outer things. And it's really interesting because one of my friends had done a little bit of research again, and finding out that strangely, geranium species for the most part around the world are kind of like legumes. So they're not necessarily recalcitrant. It does help to keep them moist and sow them right away, but essentially they just need to have the seed coat crack and to get some water in them, and then they're pretty much non-dormant. So typically we would sow these in the late summer after keeping them just in plastic again on the floor at my parents' place, and not putting them in the fridge through the summertime. Actually, you can actually see this in geraniums. The entire seed is actually the baby. So there's a seed pod in my picture here there's a little bit of green showing. So if you peeled the whole seed coat off, that's the little plastic bubble, the whole thing is just like a sugar maple seed on the inside. So yeah, they're very fascinating. Oops. Okay, so I'll do the pawpaw for 5 to 10 minutes at most, 5 minutes, and then yeah, we'll have time for questions. So pawpaw to me is one of these, it's a bit of I'm going to call it a cult native plant. It's kind of like garlic, you know, there's kind of a cult status around pawpaw. It's naturally rare, but apparently it's everywhere on Facebook. I've been blown away, especially through COVID, how many, you know, how much native plant trade picked up online. But there's so many specific Facebook groups about pawpaws. It's wonderful, but it's just kind of incredible how much seed trade there is around. And of course, you know, a lot of people have pawpaws now and understand that it can be challenging to grow, and you have to keep it moist all the time. So I kind of think that it's like woodland ephemeral plants, and you have to be patient with it the same way. And it's got pollination issues, and it's got all these different you know, challenges, but it's very enigmatic. So I do this strange thing at work where I tell people to text me if they're having a seed problem right away. So I have I'm not suggesting everyone should text me right away, but we do and help a lot of colleges and universities teach their students about seeds. So we'll send out samples, and I had collected quite a bit of pawpaw seed in 2021. It was a really big year, and one of our collaborators down in Southwest Ontario has several sources, and he has a big garden of it. So he said, oh yeah, come take as much as you want, and we could share them with folks. So one of the colleges got this, and they texted me and said, we don't even really know what we're looking at. Like, what are we trying to find? And if you've ever cut open a pawpaw seed, it is hard as a rock. It's probably one of the most interesting seed morphologies in Canada that I know of, because it feels bony. It feels, it never gets soft. Unless it's rotten. So it's very interesting. And when you're trying to look at the picture in the Woody Plant Seed Manual, you're going, you know, you would think for most seeds that the pointy end is where the embryo is. But on pawpaw, it's the opposite. So the pointy end is what shoots away from the root. So the little tiny embryo in here is nestled amongst all the folds of this brain-like structure that we're still not quite sure why it's folded that way or what it actually is doing to help the seed. But basically there's a tiny little suture at the end, and that's where the root can eventually get enough strength to push out from. But it's extremely small, and it's the same problem with the magnolia that I had, which is you start to cut them apart and you're trying to discern white from white and trying to find a tree. So I offered to help them, and they were going to try and do some tetrazoleum. And I don't know if anyone's ever heard of tetrazoleum, but we use it for really tricky species. We use it to start to figure out if we can dry things before we store them. So because the seed information database, the worldwide one with the SER or the Q seed information database, it says that pawpaw is still kind of uncertain. A lot of people will say that it's recalcitrant. Lots of growers say it's recalcitrant. I'm very much someone who tries to challenge this so that if we needed to store something for a long time, we could. And so for us to even do cryopreservation, we need to test seeds and slowly dry them and then just see if they stain red. But yes, the whole concept of tetrazoleum is that it's a biochemical reaction that turns seed tissue that's alive very red. So like fire engine red. So we use it on all sorts of really tricky species. And so we had a whole bunch of this seed in 2021. I said, hey, you know, the colleges will try it again. And so we have several books. I find it very strange. This is just a little side note. I find it very strange that the American Seed Testing Association doesn't have pawpaw in their book, but the international one does. So it's just a little oddball thing, but they have these really great photos. They show you the exact protocol for how to cut them apart and how to actually tell if it's a good stain or not. And we always feel so confident, and I always say I'm confident about things, but this one was really challenging. So trying to get the seed coat off was difficult. It was also very dangerous. I don't want anyone to cut themselves when they're doing things, so be careful when you do this. And we have these little trays. We turn them all sideways and down, and then put the tetrazolium liquid. It looks clear at first, and then it would start to react. And so you put it, we have a little oven that keeps things steady around 30 degrees Celsius. And it just, the stain wouldn't even penetrate. So it also has an oily component in the endosperm. So it was actually resisting absorbing the tetrazolium. So it was very strange to try and figure out why this wouldn't work if it's in an international standardized seed testing book. So even I was confused. But it was, again, really amazing to just start to have to figure this out, and then to finally find a couple little trees, and, you know, after all this digging, to try to get a confidence sense of where this little plant was. So I just love these little photos where when you finally get, so this is a living pawpaw tree that's stained red and alive. And that's the two little first leaves, and the two are in the first root. So lots of times I feel often as if I take some of the, you know, some of the first photos of something that most people haven't seen. But the whole point of what I want to say about pawpaw is that you're better off just sowing it and growing it in the garden right away. It's very slow to wake up. It takes usually until 7 or 8 months sometimes, I've found, of winter stratification, and then it's just very, very slow because that baby is very, very small. So give it time, and definitely protect them. Anything with oil in them is usually a food source for everything else. I mean, all seeds are food, but I do want to say that you do need to do a lot of protection of these guys. So I do love pawpaws, and I'm going to stop sharing my screen now, and I'm just going to go to, for Bill's recording, whoops, I'm just going to go to my last slide so that it's recorded. Hold on, I'm missing something. So yes, I had more, and I will take some questions, but yeah, for the most part, again, if you guys want to email me, that's just my personal email. You can find me on Instagram and all the wonderful seed things I post. I'm on Facebook as well, even though I'm not terribly pleased with the new AI that's been released. And I like to celebrate all my little babies having babies whenever we can. And I do a lot of seed collecting trips, and love to, you know, tell people about all these different things. So I hope that's good. And I apologize for rambling as I do, but I appreciate your time.
Melissa, that was great. Really, truly appreciated the presentation, and I don't know whether I'm more challenged to try spring ephemerals or I'm going to back away because, oh my gosh. We did have a couple questions, and several that surround the hydrogen peroxide. So one question is, when do you add the peroxide? At what stage is it after you've collected? And kind of what is the process that you use for peroxide treatments, and then for briefly drying out the elastomes?
Yeah, okay. So so I'll just go back through my mental Rolodex. So wild ginger in particular, I've done a lot of. So lots of times we would bring the seed pods in, and originally I used to split them all open by hand and like put them in a bowl, and then I'd rinse them and get most of the floaty bits off. But I started to learn about onion bags, because onion bags, if you put seed pods in, and they'll start to open on their own, and you can roll them around a little bit and the seed will fall out. So usually I would do some water rinses first to get down to pure seed, and then I would put them in, I mean, it's a salad bowl basically. And then it's just 3% hydrogen peroxide you can buy from a drugstore, filled so that the seeds are completely sort of immersed, and let it bubble away for about 20 to 30 minutes. And then I would strain it with a strainer, rinse it with water, and then put it on newspaper and let it just slowly dry on newspaper for 8 to 10 hours at the most, stirring it once in a while just to make sure the edges didn't get too dry, but just enough. So if anyone understand or has ever done sort of, you hear about sort of spin drying seed at times when you're doing big quantities, just to get all the surface water off. And the seed, I would watch it really, really closely, is that the seed would actually start to curl just a little bit. And once it started curling and sucking in on itself, that was usually my sign that the elizum was shrinking, or at least being reduced. But I, at that point, would get it in the bag. And the same thing for trillium and bloodroot, is that after the peroxide rinse and the slow drying on newspaper, and even having just 2 layers of newspaper on top of each other, slow drying being, you don't need to put a fan on these things. Like, you just want them to slowly emit, and I'd stir them around every couple hours, and just be enough so that they're free flowing, but you'd watch them just sort of shrink a little bit. So I hope that's a good enough description. I probably have a little video again where I'm talking to myself because I was doing it to train staff when I was going back and forth between Ontario and New Brunswick. But yeah, the peroxide, there's a lot of different, there is information out there on a lot of peroxide treatments. And if you're doing tree seed or you're doing things of a really thick seed coat, you can leave it in for, hours and hours and hours. But most woodland ephemerals, I wouldn't leave them in for much longer than 30 minutes.
Okay.
And you only really need to do it once. And then you should be good to sow within about 5 to 10 days.
Okay. It's very helpful and interesting. May have to give that a try. So it wasn't a question on the list here. It's one of my own. So you were talking about having seeds on the, let me check my notes, the basement instead of the refrigerator. So are you then, would you leave it in the basement as long as you might keep it in the refrigerator based upon it needs a 60-day cold storage? So you would leave it in the basement 60 days? Or
So just to clarify, like all of the species that I'm talking about, so I'm going to go through my brain. So Dicentra, wood poppy, bloodroot, trillium, wild ginger, Corydalis, like some of the spring Corydalis, like all of these things are kind of mature and ripe before the middle of July. So I would leave them just on the basement for a week or 2. And it really just simulates like a cool late spring night. And I'm not doing cold stratification on the basement. If I was doing, I got away from doing, and it's the same as I will tell anyone, like I got away from actually doing a lot of refrigeration stratification at our nursery because the winter, like our winter sowing conditions were just so much better. You got so much better success. I always find that, even with paper towel, people use it. I'm like, paper towel encourages mold in lots of species. And I just much prefer and think more people have better success with winter sowing than they do trying to do fridge techniques.
Right.
For me, a fridge is just a very temporary situation. Usually I want to get the plants outside and let them go through their natural cycles. And with most woodland ephemerals, you need to let them have that warm, they need to be warm and moist through the summer. So the embryo starts to grow.
Mm-hmm. Mm-hmm. Here in Indiana, I think we're starting to experience some climate change, and they've actually changed in central Indiana the zone from maybe a 5B to a 6 or something like that. So, our winter can tend to be a little bit milder, and then I'm freaking out because my cold storage outside is if I'm delinquent late January, I'm almost don't have one, it seems like. But the seeds seem to know what they're doing. I think you could read what Ray basically was talking about, pawpaws, high deer populations increasing in southern Indiana. And so the deers don't eat it, and it's replacing maple, beech, elm understory reproduction in many areas. So a good comment from Ray on that. Let's see, not a question. We love to have you and the webinar attendees in this pretty quiet Facebook group if you like. And then there was a link on there, but very great information. So I think for now that's, oh, we have one more quick question that's come through, and then we will turn it over to Bill. So do you put seeds in Ziploc dry, or do you add a little sand and or moisture?
Can you give me a species example, Sarah?
I don't, there wasn't one, but I presume that would be for ephemerals, but maybe for any seed.
So if I ever added media to a seed, just to keep them separated a little bit, there are certain species where a big mass of seeds is very unnatural. I would usually, if I kept things in Ziploc and I wanted to keep them moist, I would use some vermiculite or some usually the really coarse vermiculite is what I like to use. So it would keep some moisture around, but it would separate the seeds a little bit so they would breathe a bit better. Sometimes I would use sand. I liked sand for a lot of my alvar plants, not an ephemeral, but prairie smoke, I would always put in sand. Anemone canadensis, I'd put in sand. And again, keep them on the floor in the basement for a while until I was ready to sow them. So sometimes sand is really good with really small seeds just because it makes sowing a bit easier. And you can also let the sand just dry out, again, just enough to make sowing simple, but just being cautious of, not over drying things.
Good. Okay, perfect. All right, thank you very much. So I will turn it back to Bill to close up for the evening.
Well, thanks a lot. Really, really do appreciate it, Melissa. I appreciated the ant snack pack and also just the propagation method that you're using with your flats and the soil mixture and I also really I think this makes a lot of sense to do the winter sowing. Rather than refrigeration. It's very anecdotal, but I feel like I have seen better germination. Communities, you can also reach out to Melissa directly. She has shared her email. I'm not for sure if that got into the chat, but I will make sure that it's available on our Trello board that has been shared. That'll go with her bio and so forth. We invite you to join our Facebook group. Again, pictures and videos, et cetera, are welcome on your propagation, how it's going. We'd love to hear from you, especially about any of the community projects, your seed collecting, if you're having potting parties, things of that nature. Our next presentation will be Monday, May the 20th at 7 p.m. Eastern. And for this one, we will be having David Sollenberger with the Chicago Botanic Gardens Dixon National Tallgrass Prairie Seed Bank. So really looking for this. We haven't had anybody doing anything on seed banking specifically. So I've heard David do a presentation before, and it's really, really well done. And then, let's see here. Okay, so I think that's it for notes and everything. Thanks again, Melissa, for your informative session. I'd also like to thank Ray and Deb for helping out this evening. It was good to see you all. We'll sign off for now. Have fun growing natives from seeds. Thanks all. Bye-bye.